Hello, good afternoon, everyone. Thank you. Oh, the jingle is longer than I thought. Boom. We are good. Thank you very much for being with us. Thank you for those joining us online. So this is it. This is the anticipatory briefings where we get to hear from a scientist what they have on their radar for the next 5, 10, and 25 years. The first session is about the origins of life. We have with us a professor in Earth and Science at the University of Toronto, where she's the co-director of a subsurface science and exploration program. She's also an adjunct professor at the Institut de Physique du Globe de Paris Université, Paris-Cité. She's an expert on bullet number 1, the interface of the carbon and water cycles. Number 2, the origin and fate of carbon-bearing fluids. Number 3, deep subsurface microbiology, and also the remediation of surface drinking water supplies. I wanted to read you the list of the 11 awards she collected this past year, but she said, please don't do that. Let's save the time for my talk. But I tell you, she won more awards than the Canadian team got gold medals at the 2024 Olympics. I just want to mention that she was the 2023 American Geophysical Union Carl Sagan Lecturer, which is pretty cool. She's going to tell us about the latest research unraveling the mysteries of life deep within the Earth, and how these findings could influence our understanding of biology, evolution, and the search for extraterrestrial life in the next 5, 10, and 15 years, 25 years. Please help me welcome Barbara Sherwood Lollar.
Thank you so much. It's a real pleasure. The excitement of this event is already, I think, engaged all of us. As mentioned, I'm a professor at the University of Toronto, but also a program director with CFAR for its international research program in Earth subsurface science and exploration. And in the next few moments, what I'm going to do is take us on a quick tour of the deep Earth to show you some of the discoveries that we've made there about the very nature and foundation of life, with implications for the origin and evolution of life, with implications for the genetic biodiversity of our planet, and with implications for climate change. But in the amount of time we have, I'm going to focus specifically on the microbiology of the Earth's deep biosphere. Now, we know a lot about the microbiology of the surface of our planet, of environments, of soils, even the human microbiome. But microbes are, of course, tiny, single-celled, and invisible to the naked eye. And so when we move to consider the microbiology of the rocks and waters beneath our feet, there, as I'll show you, we are truly still at the frontier of exploration and knowledge. Context is important. The most important contextual piece of information I can give you is how recent the development of this discipline is. If you go back to textbooks in even the 70s and 80s, you'll see that when they talk about the rocks, whether that's the rocks beneath the continents or the rocks beneath the oceans, largely you'll see statements that say below a couple of 100 meters, there's probably nothing living. Why is this? Well, because we think very much about life on our planet in the context of life needing the sun's energy, in the context that all energy for life comes from photosynthesis that then feeds up through the food chains. We certainly understood that microbes might live in the subsurface, but the presumption at that time was largely that they would be surviving on buried organic food that had come from the surface, from photosynthesis. And that at some point, when that food was used up, that there would be no way for microbiology to sustain itself deep in the dark places of the ocean rocks or the continental rocks in the absence of a connection to the surface photosphere. In other words, we thought very much about life as if it was just a thin green film restricted to the surface of our pale blue dot. Just in the late 1970s, when I was still in high school, it turned out that Jules Verne was maybe correct after all. And the Alvin Research Submersible and others diving at the hydrothermal vents, taking down physicists and chemists and oceanographers, but not usually biologists at that point, discovered something deep in the dark parts of the ocean, which is shown on the right-hand side here. These were communities, macrofauna, associated with hydrothermal vents, tube worms 2 meters long, clams the size of dinner plates. And although some of these ecosystems were indeed living off of photosynthetic material that had floated down from the surface, others had at the foundation of the food chain, deep on the dark ocean floor, an entirely new type of microbial ecosystem. We found these as well in the continents, in fractures containing water, in rocks even 1, 2, and 3 kilometers deep. Again, some things were what are called heterotrophs, living off of organic matter from the surface, but the deeper and deeper you go, the more and more we saw organisms and ecosystems founded on a very different type of synthesis, not photosynthesis, but chemosynthesis, which is specifically the energy that's derived from water-rock reaction. And indeed, these are called chemolithotrophs, which simply means rock and chemistry eating microbes. How many of you are familiar with the term follow the water from the space exploration? Oh, good. Many of the space agencies, NASA and ESA included, use this term to define their search for life in the universe. And that's because, as we know, all life needs water. In fact, we follow that same search strategy on Earth. And over the last 2 decades, our team has actually been investigating the nature of groundwater on our planet and made 2 really groundbreaking discoveries. The first is that groundwater can be much, much deeper than we ever thought before. In high school, you probably all learned the same water cycle, the idea that the rain comes down, it recharges, and eventually all water goes back to the surface, hence an iconic cycle. What we've in fact now found is that even 1, 2, 3, and 4 kilometers deep, about 40% of the Earth's water cycle is actually trapped in the deep Earth and does not return to the surface on short timescales. And so, in fact, not only is that water deep, but it can also be very old, depending on where you are. It can be tens of thousands of years old, tens of millions of years old, and in some places, even 1 billion years old. And so as we have followed that water and particularly redefined what we know about the water cycle to include what we now call the hidden hydrogeosphere, then teams of microbiologists have come with us in order to investigate, follow that water, and investigate what lives in these deep, old systems, particularly in the continents. There are a number of major questions that are still being asked. There's much we do not know. One of the first ones is simply how much is there? Some estimates say maybe only 10 to 15% of what's found on the surface. Other estimates say 90% or even as much deep biosphere microbial biomass as there is on the whole surface. A very wide range of estimates. Obviously, a lot of uncertainty and much work still to be done. Other teams are finding clues to the origin and evolution of life by examining these organisms. It's a practical question right now, not did the origin of life actually occur on the surface of the planet, in a warm little pond like Charles Darwin envisaged? Or could it be equally possible that the origin of life took place in a warm little fracture, isolated from what was a really hostile environment on our planet 4 billion years ago when life first arose? We're finding information about evolution as well. In 2010, teams diving in the Arctic Ocean found organisms at the hydrothermal vent that are a genetic missing link between single-celled organisms and multicellular organisms. So all kinds of information is coming from these systems. Many of the organisms are genetically similar to what's found on Earth, but many are entirely novel. And in particular, we see extremophiles, organisms living at extremes of temperature, pressure, and salinity. These are exciting, not only in terms of changing our understanding of the full biodiversity and capacity of life on our planet, but they also have very practical implications. Coming out of COVID, we're all very aware of the issue of the transportation and storage of vaccines. Many vaccines needing to be contained under very narrow temperature ranges because if they're overheated, they no longer function. So as you can imagine, there's considerable interest in investigating extremophiles whose proteins are able to function at much higher levels of temperature. So there's both very fundamental and very applied questions being asked. I wanted to give you one more unusual example. Again, there are many metabolisms in the deep subsurface very similar to the surface, but there are some that are really surprising, and this was one of them. We've known since the days of Marie Curie that when radiation naturally occurring in the environment hits water molecules, it breaks them apart and produces hydrogen as a byproduct. But we never in our wildest dreams assumed that life had evolved to take advantage of that process. But we now see in some of these deep systems exactly that these chemolithotrophic rock-eating organisms are surviving from hydrogen produced from that radiolytic breaking apart of the water molecule. Hydrogen is a very important food source for microbial communities. And it's more than just hydrogen. We see as well that as the other half of the equation is that water molecule splits apart, it forms hydrogen peroxide and other really reactive radicals. And that when these interact with minerals from the rock, they can produce more food for microorganisms, including electron acceptors like organic compounds and sulfate. But what's really cool about this, if you don't remember all the chemical reactions, and I don't expect you to, what's super cool about this is that this is a self-contained system. It can produce all that they need, the entire smorgasbord for microbial life sustained in the subsurface in the complete absence of connection to the surface photosphere. It's a mechanism by which life can be sustained even in parts of our planet where there's no photosynthetic input. And for space, this is particularly important, because it means that on planets where photosynthesis might never have developed, there's still a mechanism by which those planets might be habitable through a process of, again, chemosynthesis by radiolysis. Over the next 5 years, we've certainly got a lot that's going on in this nexus between the surface and subsurface life. And I'm referring to this somewhat as a clash, because particularly as we are adapting to the transition related to the climate change and other major changes on our planet, we are rapidly impacting the subsurface, both in terms of extraction, whether we're taking out water, critical minerals, or energy, and in terms of injection, what we put into the subsurface, either accidentally through groundwater contamination or purposefully, by targeting the subsurface to be used for disposal of waste, or even for things like subsurface storage, for carbon capture and storage, or natural hydrogen storage for energy. And the key thing is, this can have both positive and negative impacts. I'm just going to give you one example of a positive impact, and that is that subsurface life can help us. Natural bioremediation from organisms that simply naturally exist in these environments has become a billion-dollar industry over the last 20 years. I'll just give you one example from our own laboratory. One great example, a shining example of international collaboration, was the 1987 Montreal Protocol that banned the production of CFCs because their contribution to the ozone hole. But those chemicals are still around. We have both legacy of them as well as continuing illegal production and trade. And those CFCs enter our subsurface, enter the ground, both accidentally and purposefully around the world. Recently, it's been found that these can actually be cleaned up by naturally occurring microbiology. So these are a huge potential for a whole variety of pollutants to have the microbiology help us. There are negative aspects as well. The microbiology can be working against us, particularly for some of these subsurface storage, carbon capture and storage, and others. Microbially induced corrosion or biofouling can make those projects more difficult. But the key thing is, we really are only beginning to understand the impact that we are having with all of this activity on subsurface life and biodiversity. Over the next 10 years, but starting right now, and we're probably already behind in terms of this pace, are the implications for space exploration. As I mentioned earlier, the studies that we're doing to understand much better the breadth of life on Earth is informing how we go about looking for life elsewhere. When we look even at the exoplanet atmospheres, we're no longer just looking for oxygen that's a tracer of photosynthesis. We now recognize that chemosynthetic biosignatures have to be part of our search strategy as well. Right now, the potential to actually return samples for study, either from asteroids or from Mars sample return, has very much refocused attention on what's called planetary protection. Again, there's been an international treaty since the 60s that the space agencies have abided by that allow us to develop protocols both to ensure that the missions we send out do not send terrestrial contaminants out onto other targets, but also as we bring these samples back for the first time, and we're really entering into that generation, as we bring them back to study them, that do we not contaminate Earth with what we bring back. And then in particular, an enormous rise right at this moment, and certainly accelerating over the next 10 years, in terms of a massive change in the way we do space exploration, the development of private sector exploration and resource-focused exploration. And how will they follow the same planetary protection treaties that the agencies do? I'd like to refer you tomorrow to a really important session on governing space that I think will pick up some more on those themes. Over the next 25 years, but again, what I'd like to emphasize here is not in 25 years, starting right now and continuing to accelerate. Climate change is, of course, in this sector between surface and subsurface, also the great disruptor. We're all used to these pictures of increasing, in this case, need for critical minerals around electric vehicles, increasing in injections related to shale gas. I could have shown pictures of increasing need for water. All of these rapidly increasing crisis points in terms of our relationship with the planet as we search for energy, as we try to adapt to a green energy transition, look for critical minerals, need more groundwater, or once again, as we use the subsurface more for drilling or fracking or storage. Again, the impact on subsurface life is still largely unknown, despite the pace of these activities. It is more than time for international action on the subsurface. We have laws of the sea. We have a treaty for Antarctica. We have the outer space treaties committing to peaceful needs. Probably more than enough time for us to think strongly about the need for international agreements around the use of the subsurface. At the moment, it is a hodgepodge of complexity. Mineral rights, water rights, many of them local or regional only. There's urgent need to develop, at the very least, international best practice and hopefully policy and guidance. How do the global stakes that we all share as citizens on the Earth in biodiversity and water, in equitable resources, balance out local needs and local sovereignty? And again, we need to do this not just for Earth, but in fact with implications for the whole solar system. And so just to end, the key point here, and this really ties into the overall theme of GESDA this year in acceleration, is the rates of change are driving the train of a planet's future and the interaction between the surface and the subsurface faster than we can develop the research to understand, test the possible solutions, and launch collective actions and policy to guide it. And just to finish, really, we cannot protect and manage what we do not understand. Subsurface life is truly a frontier, and yet I hoped I have given you in these few moments a sense that it is intimately related to our and our planet's future habitability. Collective global action at all levels is needed, but often, whether I'm speaking with other scientists, with policy, or with my neighbors, this knowledge that there's life interacting with us within the deep parts of the Earth is still in many places in its infancy. Allowing people to understand more about it, and I hope I've been able to do that in the last few minutes, is the first step to having us step up to think about how we will manage these precious resources and the responsibility that's associated with them. Thank you.
Thank you very much, Barbara. So we are going to have 10 minutes for questions. Please raise your hand. I guess we have some microphones going on. So the fastest hand was here, the second fastest, but she was the closest.
But I've got the microphone.
Good afternoon.
Thank you. That was fantastic. Elaine Franz from Flow in Action. You said something really powerful right at the start around what we didn't believe in the 1980s. And I've been in rooms and facilitated meetings where people believe the science, but they don't believe we can get to the solution fast enough. How can we, and it goes to your very last point also, how can we reconcile this on a daily basis? How do we get people to believe now so we don't end up in 25, 50 years and go, oh, well, what we believed in the 2020s was obviously crap. How do we do that? What piece of advice can you give us to get there?
Well, first and foremost, it is important to understand that our understanding evolves. I mean, we all know this. I mean, the fact that we didn't understand something before didn't mean we were crap. It just, things evolve as you explore. And so this is a journey of exploration to a certain extent. We had not explored the subsurface of our planet. But to relate back specifically to your question, I think it's about doing everything we can at every level of society and every demographic to broaden open-mindedness, broaden the imagination. I spoke of Jules Verne, but I'm often inspired to think back at those narratives, the way in which those literature and philosophy about the imagination is an extremely important part of this. We tend to think about scientists as test tubes and laboratories, but in fact the ability to communicate, to spark the imagination of everybody. I mentioned my neighbors. My neighbors do actually have to listen to me sometimes. But to specifically answer your question, I think it's events like this that open the doors to as many people as possible from as many different disciplines, because it isn't a unique directional communication. It's not about science laying down the law. It's about listening. It's about communicating. Much of our research has been informed by important questions coming from communities. So I think it's a wonderful way forward.
You already have the name of a superhero cartoon you could do, the Extremophiles. I thought that was a really cool name. We have so many questions, Barbara.
Yeah.
Let's try to get as many.
I'll try to make it quick. Yeah.
Yeah, thank you so much. I entered this room a bit mindlessly, and now I'm all inspired. So thank you so much for this. I think I have a somewhat philosophical question. I think we all agree on what non-life is, rocks and gas. We agree on what life is as well, whether it's species or plants. But can you elaborate on when does life actually starts to be? When is this threshold passed? And maybe to extrapolate a little bit, what does this threshold imply for the future of life? People talk about artificial life. What are your thoughts on this? Thank you.
The key thing, there's no way I can give a short answer to that one, except to say, in fact, we don't all agree. This is a huge topic of conversation, even amongst scientists. How do we define life and non-life, and certainly amongst philosophers? And it very much feeds into space exploration and research as well. So with apologies, impossible to give a short answer to that one, but I'm happy to pick it up and talk a bit more with you afterwards.
It's a fair question in a session called The Origins of Life.
Absolutely.
Sir.
Thanks a lot for this great talk. Two questions. First, a nerdy question. What's the distribution between bacteria, archaea, and eukaryotes deep down? And the second question, any worries about long-term nuclear waste storage down there, if there's all that life down there?
I'll start with the second one. The first and foremost part is there isn't life everywhere. This is one of the things that we're trying to address and understand. And the places that have successfully put in place radioactive waste around the world have done so with decades worth of research showing that they're putting them into places, usually with very low porosity, tight systems in which there isn't water and typically isn't life. So the research has been done and is being done with enormous rigor to address these questions. In fact, some of the nuclear research agencies have funded teams all over the world in this kind of thing because they want to do it right. Secondly, in terms of the distributions, this is where we're really at just now, trying to understand how we move from one distribution of life to a different one in the subsurface. So in particular, you asked about eukaryotes. So we do have a little bit of information about that with work by a colleague by the name of Gaetan Borgini, who is an expert in nematodes. In work in South Africa, we're actually able to identify that you find the nematodes, which need to be in the presence of a little bit of oxygen, but you only find them within waters that still show that ancestral connection to the surface. As soon as you get into the ones that are cut off on long time scales, most of the eukaryotes go away. In fact, to date, I'd say all the eukaryotes go away. So we're defining that geography of the deep subsurface. But again, it's still early stages, and understanding the controls on it is really a compelling question.
Hello, thank you for your talk. I just have a quick question. Actually, I'm not sure if it's going to be quick, but how do we protect our life, new life, from old life and the origins of life in terms of, because you mentioned that there's the water down there may be millions of years old, billions even, and there may be life trapped there that we haven't been exposed to, such as potentially dangerous pathogens. How do we mitigate the risks and protect the security of the current human population life on the surface while continuing to explore and expand our knowledge on this topic?
Great. I can actually give you two quick answers to that. One is very carefully. Planetary protection is something we use on this planet as well. And we do it not just to protect ourselves, but also to protect the results. Right? Well, the last thing we want to do is contaminate a sample. So it's a very careful process. But secondly, the important thing to understand is life down there is we've probably arisen from that life. And so although we do obviously scan for pathogens and make sure that there's nothing of that nature, to date, that's not anything that anybody has found. Typically, these are more similar to the kinds of organisms that are in your stomach right now, typically. They're anaerobes. And to date, there have not been any results showing any kind of dangerous organism coming out of these systems at all. But that doesn't stop everyone from being incredibly careful, because best practice prevails. You do best practice not because of the probability of an adverse outcome. You do it because it's the right thing to do.
Thank you very much, by the way. All of you asking questions, you get this first thing you said, like a philosophical question, two questions, one question spoken slowly. Thank you for like the warnings. One question.
Okay.
If you can.
And I'll answer quickly, I promise.
We still have 2 minutes. We're pretty good.
Oh, okay.
Don't rush too much.
All right.
First of all, thanks so much for the talk. That was very inspiring. My question is about the evolution of these lineages. So you mentioned that they're quite genetically distinct from the terrestrial lineages. What do you know, if anything, about the most recent common ancestor, when these lineages diverged, and which perhaps was ancestral compared to the terrestrial lineages?
It's a huge burning issue at this point in time. The key thing is not, some of them are very much related to what we find elsewhere. For instance, if they look like something we've seen before, they typically look like something we've seen at the hydrothermal vents in the deep oceans. So clearly there's a degree of commonality that suggests not any evidence for a second origin of life. So they're all DNA as we know it. They are, where they differ is in typically metabolic functions, like that radiolysis, different ways of gaining energy. The question of evolution is a really, really important one, because previously, I said what things we did in the past. Previously, there might have been a tendency to, find a rock, find a microbe on that rock, and assume that that microbe's the same age as the rock. Happily, we've gone well beyond that now, because again, this is simply not best practice. And what's embedded in best practice now, both on Earth and for NASA and ESA, is an understanding that it is a three-part problem. You need to obviously describe the rock or the environment in which you found it and how old that rock is. You need to describe the life that you found. But you really have to look at the water, because that is the last mobile phase that moved through. And to be conservative and careful, and we must always be so, then the life could be as young as the youngest drop that went through the system. So if you're reading a paper that just finds a rock and a microbe and that's all they tell you, be cautious. Best practice would involve looking at all three, and only once we get those timings nailed, and we really haven't on a global scale yet, can we begin to say when what we see here and what we saw there diverged. When we get those times right, then we can make the evolutionary arguments. And we're not there yet. We're still pushing very hard for those timescales.
There were some headlines coming out a couple of days ago about some 2 billion years old microbes being found. What you're saying is that that might be the age of the rock, not necessarily the age of the microbe.
Absolutely.
We have time. I have 15 seconds remaining there, so it has to be a very quick question and a very quick answer. Sorry, guys.
Oh. But I'll try. No, I'm so amazed again about those, what you said at the beginning, that before 1980, we were not able to distinguish what started. But you mentioned, of course, Jules Verne, but when I think about all the geologists' work, the famous Jacques-Yves Cousteau, the Picard family, which is from Switzerland, would you dismiss everything they've done, or how would you characterize their work prior to 1980?
I met Cousteau when I was 8 years old and have been inspired ever since. No, they were looking at the liquid ocean. What I'm referring to is once you step on the rock, so deep into the rock of the ocean floor. So all of that work taking place in the 60s and the 70s by Picard and Cousteau and all of the other explorers is in the ocean water itself. There are many important geologists, yeah, Hess and the others, but there wasn't, before the 60s, before the development of the big ocean drilling rigs like the IODP and some of the others, there was not the ability to drill into the ocean crust deeply. So that's the distinction. When I'm talking about the deep biosphere, I'm talking about is rock hosted. So not what's in the deep oceans swimming around, but once you hit the ocean floor, whether that's at 100 meters deep or deep at the bottom of the Marianas Trench, what happens below that in the rock itself is the talk that I was focusing on.
Thank you very
sense?
much.
Okay.
That was Barbara, Sherwood, Lollar. Big round of applause.